So now I need you to turn to page 99 in your textbook as you are following along on the good PowerPoints, hopefully not necessarily the ones that I am shaking and shivering around on now. Let's talk about HIV infection. Of course, we know that HIV stands for Human Immunodeficiency Virus, and it is the virus that causes what we used to call AIDS, acquired immunodeficiency syndrome. They're kind of getting away from that term now, and they refer more to HIV infection. And rather than full-blown AIDS, they usually will refer to it as end-stage HIV. But you'll still hear the AIDS virus talked about. HIV is a viral illness. It at one time had a very high mortality rate. When it was first discovered in the early 1980s, 1981, 1982, generally the person who had it was not diagnosed until they were considered to be in end-stage HIV, full-blown AIDS, where they had a variety of opportunistic infections taking over. Now, thankfully, 30 years later, we have reached a point where people are considering HIV to be more of a chronic infection that can be controlled with medication rather than a death sentence. So thankfully, things have changed because of medication. There is still considered to be a very high-risk population, and unfortunately, the first generation that was faced with AIDS when it was a death sentence really took every effort to bring the illness under control, to encourage medication testing, to encourage safe sex practices, and they really got the word out. Nowadays, now that it's considered more of a chronic illness and there is medication to treat HIV, people are getting more complacent. We're not engaging in the kind of safe sex practices that curb the infection early on. The high-risk populations still include gay and bisexual men, intravenous drug users, people who are in prison because there still is same-sex activity in prison, as well as IV drug use. The female sexual partners of people in the 3 high-risk groups that have already been mentioned are at high risk, and children born to mothers at risk are still considered to be vulnerable populations, although, as we're going to see later, thankfully, medication and other practices has really decreased the chance of children testing positive for HIV virus. HIV is diagnosed by blood test most of the time. If you look on pages 98 to 99 in the textbook, it explains the life cycle of the HIV virus. And there are 3 main stages to HIV. The first stage becomes entry to a target cell. And if you watch that previous YouTube video that is just talking about flu and other viral illnesses, you kind of see how a virus gains entry to a target cell. But what makes HIV virus so deadly is it is not a regular virus that eventually grinds to a halt on its own, and as the immune system takes over. This retrovirus is able to use its own RNA to transform the DNA of a cell into a copy of the virus's own genetic code. And once it does that, it just changes the cell's genetic machinery completely. Once that's done, the virus can grow, the cell ruptures, and new virus particles, virions, as I've mentioned, can be released and target other cells in the body. Now, the main target of the HIV virus is a T lymphocyte, a thymus-derived lymphocyte, which is one of the many types of white blood cells we have. The surface of the T lymphocyte is coated with a kind of a glucose protein combo called CD4. Now, that completely coats the surface, and HIV, the virus, attaches to the CD4. And when it's able to wipe out those cells, the CD4 count becomes very low. Those T lymphocytes are destroyed. So what the test used to assess the function of the immune system in the presence of HIV is called the CD4 count. So in the average healthy person, the CD4 count is anywhere from 500 to 1,500 CD4 cells per microliter of blood. But once HIV occupies those cells are destroyed. When the CD4 count reaches 350 cells per microliter, it's considered necessary to have started drug treatment. And when the person's CD4 count drops to less than 200, that's considered to be full-blown AIDS or end-stage HIV, because that is when these various opportunistic infections, I think there are 25 or 26, can start to take over because of the damage to the person's immune system. And that's considered to be end-stage HIV. So what we're aiming for is a CD4 count that is within normal limits, 500 to 1,500, and a viral load of as low as possible. And with the use of these medications, sometimes the person can reach a viral load that is non-existent. You can't even find any viruses. However, that is because of the medication. And when a person stops taking the medication, the viral load will start to multiply again, and fairly quickly too. So it's very important to tell our patients who are being treated with HIV meds, antiretrovirals as they're called, that they cannot stop. It is not a cure. It just keeps the virus under control. So on page 100, we start talking about antiretrovirals. And there's a table that gives the various stages of antiretrovirals, and there are a number of them. A retrovirus is a virus that has messenger RNA that codes for the HIV polyprotein. That means that when it takes over a cell, it changes the cell's DNA to its own RNA, and it will start rapidly reproducing HIV proteins. So what we need are antiviral medications that interfere with the ability of a retrovirus to either reproduce or within the cell, or to replicate by breaking open the cell and spilling virions. Now, early in the days of HIV treatment, we had two main types of antiviral drugs. They were called reverse transcriptase inhibitors, and these fell in the categories of NRTIs or NNRTIs, and you are not going to have to memorize which drug is an NRTI and which is an NRTI, but you do know that they worked early in the life cycle of the virus to stop that changing the DNA to the virus RNA. Then they came out with another category called the protease inhibitors that acted later in the viral life cycle to stop it from stop an enzyme that allowed the replication of the virus. Now we have other drugs. We have entry inhibitors, fusion inhibitors, HIV integrase strand transfer inhibitors, and then combinations of these. And I have what I would ordinarily give you as a handout, but I've put it on, I'm going to post it to the wall so that you can look as we're talking about these different kinds of medications. You can follow along in your book and refer to this picture that I'm going to put up on the PowerPoint. Sorry, I'm shaking this thing all over the place.